So Tom, let's talk about some promising experimental therapies. Let's focus on both things that are in at ASCO 2015 as well as those that interest you in the clinic, and put it in the context of we have our frontline approved therapies, we have our second line approved therapies. How are these experimental therapies going to fit into our paradigm?
Absolutely, thank you. So we really have what I would consider 3 molecules that are getting a lot of attention. 2 of them are getting attention at this meeting here, but the first one, which I think is the closest to potential approval, regulatory approval in the United States, would be the METEOR trial, which we did not see any data from here at ASCO 2015, but is expected to report out hopefully within this calendar year. And that was a phase 3 trial of the novel drug Cabozantinib, and it was versus Everolimus as a second-line therapy. And Cabozantinib being a kind of pan inhibitor of different tyrosine kinases, the ones that we think are important are going to be c-Met and VEGF. So it does inhibit VEGF, in that for the proponents that feel that you need to constantly inhibit the HIF pathway with VEGF inhibitors in multiple sequences, that appeases them. But then c-Met being a pathway, that c-Met may be involved in angiogenic escape, maybe one of these pathways of resistance. Certainly there is some thoughts that it may have a role in the papillary subtype where there can be c-Met abnormalities. Has been exciting to see that drug. And the drug was in phase 2 trials. Now we have the phase 3 trial. Many of us have experience using the drug and have seen favorable outcome. The issue with the drug is toxicity. It brings in a toxicity profile that is unique to it, that needs to be managed. And some of this is in the way of fatigue, right, but then sharing some of the same toxicity challenges that the other VEGF TKIs have with it. So that is one exciting drug that will be on the verge of data release, and we'll see if that becomes positive. And then the 2 other ones are actually combination strategies. There is the Levant Nib plus Everolimus trial, which was actually a 3-arm trial. It was a phase 2 trial, and I was one of the authors of that, and our colleague Bob Mozer will be presenting that as an oral presentation here at the meeting. And in that particular trial, patients were randomized in the second or third line setting to receive either the Levant Nib plus Everolimus versus Levant Nib alone or Everolimus alone as a therapy. And the Levant Nib is also known as E7080 in its early development age. And Levant Nib is also one of these VEGF inhibitors that also inhibits FGF. And FGF, fibroblast growth factor pathway, may be a pathway of escape and involved in resistance. So coming in with a drug that not only inhibits VEGF, but then also potentially one of the escape mechanisms is exciting. And based upon what's been published about this particular trial that we'll hear more about from Bob, has been that the combination seems to be very exciting in its PFS, a PFS value in excess of 12 months, almost a 14-month PFS that is greater than what we've seen with single-agent Everolimus or single-agent Axitinib in that setting. So the ability to do that without excess toxicity is exciting. And then finally, I think the third drug is this Cerulean drug. It's the CRLX-101, which is a novel formulation of a campothecan chemotherapy drug, which may be ultimately involved in HIF production, but also may be cytotoxic in and of itself. And it's combined with bevacizumab, and that is in the third and fourth line setting compared to standard of care, so dealer's choice in the standard of care. And what we've seen so far, I was involved in the phase 1 trial, has been a high level of activity as measured by progression-free survival, again, in the 10-month or excess range, which is far greater than what we've seen traditionally in our second line and third line setting with our currently available drugs. So there is ongoing research with these novel strategies to hopefully drive a PFS. And the big question, Bob, is going to be what we've already alluded to earlier, is how do we take the IO drugs, what we're going to expect to read out from nivolumab and other drugs in the second line setting, how are they going to play out with our current drugs? And then how, if some of these other trials are positive, how do we all incorporate them all? When endpoints are different, we're looking at PFS as a main endpoint for some of these new trials. And we've got nivolumab and the immuno-oncology drugs, which are going to be overall survival.
So David, let's just complete this segment by adding one other agent, that's delantercept, which has been reported out. So describe that drug, kind of the results of the DART trial, and and put that in context for us.
So Tom was just talking about angiogenic escape, which is sort of a main problem for our patients. When you make a tumor hypoxic, from almost the minute you start treatment, the tumor is struggling to find ways to survive. What are the pathways that allow it to survive that hypoxia? He mentioned several. He mentioned c-Met, which is a major pathway. He mentioned FGF, which is another pathway. We're learning about these pathways from preclinical experiments where we look at human tumors in mice and see, what's driving this resistance. And I think the hope for a variety of these newer combinations or agents that block multiple pathways is hopefully it's a little bit more rational design than we've had before, less empiric about just putting two drugs together that might work but not based on strong preclinical data. We've struggled with combinations to this point. All the combinations of the FDA-approved agents thus far have led us, we've gotten early encouraging activity and then toxicity problems when we get to phase 3. But now this Delantercept, which targets a pathway that is thought to be important in the maturation of the tumor blood cell. So when you block it, it turns out that it's a very complicated process. It's not just about blocking VEGF. You have to block other pathways to control tumor growth. Rupal Bhatt, who's a scientist in our group, has studied this molecule in her mouse models, and it's one of the best at preventing angiogenic escape that we've tested, admittedly in preclinical models. And what the DART trial early on has suggested, which I think is encouraging, is we're seeing, compared to historical controls, encouraging activity, but you have to be careful with comparing to prior data. It's not randomized data yet, but also tolerability, where we've struggled with these combinations, as I mentioned before, getting them safely into patients. This combination seems safe in the early going, and hopefully the randomized trials, which are essential, will show a benefit. But it's designed a little bit more rationally than our prior, and hopefully that will be the trend going forward, is that we use our science to design better drugs, better combinations.
So what sounds interesting is that we have agents like Cabo that are still opportunities for VEGF blockade and MET blockade,
right.
Different pathways. We have the DART trial where we're thinking about strategies that still look at angiogenesis. Mm-hmm. And I think we have to remember that the clear cell renal cell carcinoma is a VHL-driven tumor, right. And that is a pro-angiogenic tumor through its somatic mutations. Mm-hmm. And we can't forget that even while we develop other strategies. And even CRLX-101 has part of its mechanism of action through inhibition of HIF. So I think it remains an exciting time in kidney cancer. The challenge, though, is, again, as we enter, and I'd be interested in just a few moments, as we enter kind of the checkpoint inhibitor era, is how to integrate, sequence, combine in a way that's going to have an outcome that's positive for our patients, not just for the trial. Right. And it's one thing to develop and have a positive trial. It's another thing to integrate kind of the sequences of where we're going. But let's turn